Translation. We just reviewed transcription, the first step of protein synthesis. Now we're going to talk about translation. Translation involves reading the mRNA strand and using it to build a protein. Where does this happen at? That is correct, at the ribosome. So, how does mRNA actually code for proteins? We already figured out we take a DNA, we can produce an mRNA strand. Everything looks good here. I've got my matches. My T's are matching with A's, and my A's are now matching with U's. C and G are still matching together. But then I got to get from that message to a chain of amino acids. That's what MET, ARG, VAL, ASN, those are short for amino acids. Methionine, arginine, valine. So you're going to have several amino acids, and I'm going to give you a chart. You will not have to memorize them. How can you code for 20 amino acids with only 4 nitrogen bases? Okay, good. So, in other words, you're going to need more than one nitrogen base to code for an amino acid, right? Because C couldn't code for one amino acid. Otherwise, you'd only have 4 amino acids. So we're going to have to combine these letters. How many letters do you think it takes to code for one amino acid? Yeah. 3, that is correct. 3 letters to code for one amino acid. And since there's 4 different letters, I can put them in many different arrangements. That's how I'm going to get all 20 of my amino acids. So, mRNA codes for proteins in triplets. That's the 3 letters. Here you can just barely see that was an A right here under the mRNA. AUG codes for methionine. CGU codes for arginine. GUA codes for valine, and so on. Every 3 letters is going to code for one amino acid. We call that an mRNA codon. A codon is the 3-letter word that codes for an amino acid. So, cracking the code. This is going to be some information about scientists. Remember Watson and Crick? Crick was the guy who determined the 3-letter codon system. He was the one who figured out that it takes 3 nitrogen bases to code for one amino acid. This is how it would look if we were going to write a sentence. Why did the red bat eat the fat rat? It's a sentence made out of 3-letter words. All the words in the sentence are 3 letters. That's how your DNA is written. Here's the codon chart. One thing I want to point out is that every codon chart that you see, it will tell you if it's an mRNA codon chart or a tRNA codon chart, and I'm going to talk about what that means in a minute. This particular one is an mRNA codon chart. You're going to always have this chart available to you, so you won't have to memorize which letters code for which ones, except one code that you need to memorize, and that's this one. AUG. AUG you need to memorize as a start codon. That's the only one you have to memorize. Its actual amino acid is methionine. Everybody say methionine.
Methionine.
When you see that, that means start making a protein. Methionine is the code that tells the ribosome I am to start building. You also have stop codons. You don't need to memorize those. But when you see them, they'll have a stop next to them. That means stop building a protein. What would happen if you were building a protein and you had a mutation, a change by accident, that created a stop codon? The protein would be incomplete. The ribosome would stop making that protein at that stop codon, and you wouldn't have the rest of your instructions for that protein. You would stop right then. So mutations that produce stop codons are detrimental. No, they would be 2 separate proteins, because after the stop codon, you're done making that protein, you're going to start a new one. They won't be connected.
Oh, so there's always going to be AUG to stop.
Right. Every gene that codes for a protein is going to start with AUG. It's going to end with one of these stop codons.
I mean, what if you don't have a stop?
you would never stop making that protein. It would get really, 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 really big, right? Just keep going and going. I don't think that happens. Yes. So UAA codes for stop. UAG also codes for stop.
So stop means the same thing?
Yes, stop means the same thing. You stop making that protein.
Is it the same protein?
Yeah, it's the same protein.
They could be on any protein. So every single protein your body makes
Starts with AUG.
starts with AUG and ends with one of these three. Okay. I'm going to show you how to read this chart. This might be the hardest part when you start taking your test and reading a codon chart. But once you get it down, it's not that bad. Your first base is on the left. So do you see this pink stripe right here? That's where I'll find my first base. Somebody give me a random codon. Okay, CUA. I want to find CUA. My first base I look for on the left. See where it says first base? I find C. Got U, C, A, G choices. I find C. What's my second base? U. Second bases are along the top. So I find U along the top. Now where C and U meet, I have 4 choices. And I'll box them in for you so you can see. Here's C. All of these are C. Start with C. Here's U. Where C and U cross, I have 4 choices. See that? My third base is over here on this side. What's my third base? A. So I would find A within my C box. Here it is. CUA. Where all 3 overlap. That's the amino acid I'm choosing. It looks kind of easy, right? But what you're going to see are codon charts that just have these words in them and do not have the cheat sheet letters. So don't think that you're going to look on this chart and just find it by the letters, because in your charts that you might see, there will be no letters here. They'll just be the names, and you have to actually figure out which one it would be. We'll practice with that. How are codons matched to amino acids? So in other words, when you have an mRNA strand, it's at the ribosome, you're ready to build a protein. What is the mechanism by which the mRNA strand is read and amino acids are attached to each other? That's what we need to figure out. You're going to have a molecule called tRNA, transfer RNA. Here's where he comes in. Transfer RNA has amino acids attached to it. One amino acid is attached to every tRNA. Let me say that again. One amino acid is attached to every tRNA. Every tRNA matches an mRNA codon. So here's what it would look like. Here's my tRNA match for AUG. UAC matches AUG. UAC matches AUG. So there's the match. And this is my tRNA. He already has an amino acid attached to him, the correct one for that code. Here would be the next one. Here's another tRNA. Happens to match this one. CGU matches GCA. This tRNA has an amino acid attached to it. Now, these 2 amino acids are going to be linked together. And again, GUA matches CAU. This is another tRNA molecule. It has an amino acid attached to it. You see how I'm building my amino acid chain? Who is responsible for bringing amino acids to the ribosome? Yes?
tRNA.
tRNA. Here's what tRNA looks like. Guys, this is the hardest part about protein synthesis. Okay? You have an RNA molecule, but look, see these letters? This is an RNA strand. You got G, C, G, G, A, U, U, A. You guys see that, right? It's an RNA strand, but look at its shape. It's not just a long linear strand. It has been folded into a special molecule. That is tRNA. It has a place where an amino acid attaches to. Right here, on the 3' end of the RNA. So I'm going to attach an amino acid. If my amino acid is right here, what will this part be matching up to? The codon. If my amino acid is up here, what will this part of my tRNA be matching to? The codon, just like here, guys. It's upside down in this picture. Right here. Here's my amino acid. This part of my tRNA has to match to my mRNA codon. This picture, it's going this way, and in that last picture, it was facing down. Oops. Okay. We call this the anticodon. So we have a DNA triplet, we have an mRNA codon, and we have a tRNA anticodon. The anticodon matches the mRNA codon. We have a tRNA for every possible combination. Does every tRNA match every mRNA codon? No. No. We have a specific tRNA for every possible combination of 3 letters. I know you remember this picture. You remember this picture?
Horribly.
The ribosome? And we learned about 70S ribosomes and how their sizes were different in prokaryotes and eukaryotes. And how a ribosome has 2 subunits. Guess what the smaller subunit is? It is RNA. Guess what ribosomal RNA is? Yeah. Ribosomal RNA is the part of a ribosome, the smaller part of a ribosome. That's why it's called ribosomal RNA. It takes RNA to build a ribosome. Ribosomes build proteins. So we need RNA, ribosomal RNA, to build a protein. We need mRNA to build a protein. We need tRNA to build a protein. These guys are all of a sudden becoming very important, right? VIP. Yes, this right here, this is
because it's
a part of the ribosome. See, and you guys thought I was just being cruel making you learn that a ribosome had 2 parts. All right. Building a polypeptide. What is a polypeptide, Benedict? And what do we use to make peptide bonds?
Protein.
Maria? Amino acids. Building a polypeptide means building a protein out of a chain of amino acids by doing a dehydration synthesis reaction to join together amino acids. If you guys want to sound really smart, you just tell that to your friends. Next time you guys are enjoying some chicken, be like, man, this polypeptide tastes so good. You know how this is made? Your friends are going to be like, what? You're going to be like, yeah. Dehydration synthesis reaction coming in with some amino acids. That's good chicken. Oh, wait, there's more.
I don't want to eat chicken.
If it's fried chicken, it probably has lipids on it, right? Yeah. Fatty acids. Triglyceride. Glycerol.
You're done.
All right.
Yeah.
Here we go. 3 parts. There are 3 main stages for building a polypeptide. First stage is initiation. And no, this is not a hazing to a new molecule at a fraternity. But initiation brings together mRNA, ribosome subunits, and the initiator tRNA. We got to get all these pieces together in the right place, right? We need both ribosome subunits hooked together. We need a tRNA. We need the mRNA. We need all those things to come together somewhere so that we can work together to build a polypeptide. Elongation. Oh, it looks familiar, right? Like transcription. Elongation is where you're adding amino acids based on your codon sequence. Who is bringing the amino acids? tRNA. I knew you had it. tRNA is bringing the amino acids to the party. BYOAA. Bring your own amino acid. Okay, termination. This is the end of the codon. How do I know when to terminate? The stop codon. Let's take a look at this picture. What do you think this smaller subunit is?
RNA.
Good. What is this green line right here?
mRNA.
mRNA. What is this UAC right here?
It's
a start.
It's the start codon.
Isn't it the end? No.
It is a start codon, but I'm pointing to the one on the tRNA molecule. What do I call the match on the tRNA molecule? The anticodon. What is tRNA made out of?
RNA.
RNA strand, good. What is this MET at the top?
Amino acid.
An amino acid. Check out the picture, guys. mRNA is sliding in the ribosome. tRNAs are coming, and if they match, their amino acids will attach together. Then this one is released. After the amino acid is attached, the tRNA anticodon is released. And look, I start to build a chain of amino acids. And then my next tRNA comes, and oh, he matches. So I'll connect that amino acid. And so forth and so on until I've gone all the way down my mRNA molecule and I've read it completely with tRNA molecules and attached adjoining amino acids in a long polypeptide chain. Can you tell the story?
Once upon a time,
Once upon a time,
there was a DNA.
there was a DNA molecule right here in blue.
Made a transcript. And then it was mRNA.
What is this
It
thing?
was the most beautiful.
That
thing.
is.
Beautiful transcription strand in the lab.
RNA polymerase came along and separated what?
The DNA.
The DNA double helix. And then what started to form?
mRNA.
An mRNA strand. Then what happened to the mRNA strand?
They took out all the unnecessary introns.
Got
Introns were taken out. And then exons were spliced together. And what is this?
Is that the front? That's
It's either the cap or the
Good.
poly A.
I need a 5' DTP cap and a poly A tail. Now it's processed. Now that my mRNA is processed, what happens? It leaves the nucleus through what is this?
The nuclear pore.
A nuclear pore. Once it gets out of the nucleus, it meets up with what is this? rRNA and another protein that together form what molecule?
A ribosome.
A ribosome. Very nice. At the ribosome, what is this?
A tRNA.
tRNA. And what is this?
The anticodon.
The anticodon. The tRNA anticodon is matching up to the mRNA codon. And then, what is tRNA bringing to the party?
An amino acid.
Amino acids. And then after I'm bringing the amino acids, I'm hooking them together to form
Polypeptide chain.
polypeptide chain.
Which is a protein. We got it.
You got it.
Figured it out.
I want to show you just really quickly. There are a few differences in how this happens between prokaryotes and eukaryotes. What is a prokaryote?
A cell without a nucleus.
A cell without a nucleus, and who would be our example? Bacteria. In prokaryotes, number one, if they don't have a nucleus, their DNA is not enclosed in a nucleus. It's in what we call a nucleoid region. It's in the center of the cell, okay, but it's not protected by a membrane because prokaryotes don't have membranes. So, if it's not protected by a membrane in a nucleus, good. They do not have introns. Here you go. Prokaryotes. They're in the cytoplasm. The DNA is not in the nucleus. In prokaryotes, they actually have a circular chromosome. Looks kind of like a donut. Our DNA is linear. Strands. And our DNA is wound around histone proteins. Remember the histone proteins? Okay, we don't have that in prokaryotes. Translation in prokaryotes is sort of interesting. It happens at the same time transcription happens because you don't have to move it to another part of the cell. You can do transcription and translation all together. You're still going to have ribosomes. Prokaryotes do have ribosomes. One of the few organelles they do have. It is not a membrane-bound organelle. Remember, it's an RNA and a protein subunit together. It's not a membrane-bound organelle like a mitochondria. So you have ribosomes building a protein, and you can have transcription going on at the same time.